So continuing with your chapter on digestion, we are moving to the last part of the chapter, that is food test. So before we go into food test, just a recap of the chapter. That means your digestion process starts at the mouth, moves into the stomach, from the stomach to the small intestine, then large intestine, and finally it is removed through the anus. So in mouth, what happens is the food is broken down into small pieces and it mixes with saliva with the enzyme salivary amylase. In the stomach, the food mixes with the gastric juice, that is hydrochloric acid. In the small intestine, the food gets mixed with bile and pancreatic juice. Also, the food is absorbed. Undigested food passes to the large intestine, and finally from the large intestine it is removed in the form of feces through the anus. So when we talk about food test, you already know that food can be classified into various groups like carbohydrates, fats, proteins, etc. So for carbohydrates, the food can either be glucose or starch. Glucose can easily be absorbed into the bloodstream. Starch, it has to be broken down into glucose and then finally it is utilized by the body. So generally when we are testing whether a food has presence of glucose or not, what we do is we are going to add Felling's solution. Felling's solution is blue-green in color. Then we are going to heat the mixture and slowly we are going to heat it so that the mixture does not get boiled, but rather till it starts to produce small bubbles. And then in the end, what you will observe is formation of red precipitate, which proves that the given food sample contains glucose. Similarly, for starch solution, what we are going to do is the given sample, we are going to make a solution and boil it. After boiling it, we are going to allow it to cool and then add iodine. When we add iodine, the color turns blue, black. This indicates the presence of starch. For fats, it is quite simple. Just rub the given sample on a piece of paper and view it under light. The paper becomes translucent. So when the paper becomes translucent, it means the given sample contains fats. For proteins, it's also quite simple. Take a hard-boiled egg, put it in a test tube, add concentrated nitric acid. Be very careful when you are using concentrated nitric acid. Then we are going to boil the mixture. After boiling it, we are going to allow it to cool and then add ammonium hydroxide. Now what happens is the boiled egg is white in color. After adding concentrated nitric acid and boiling it, allowing it to cool and then finally adding ammonium hydroxide, we get orange color. This orange color indicates that the food sample contains proteins. So these are the experiments which I just told you to determine whether a food contains carbohydrate, it is either glucose or starch, fats or proteins. Next, we'll be moving into experiments on digestion. So what we're going to do is, first we're going to find the determinant of enzyme salivary amylase. What we're going to do is take starch solution. Simple, we're going to test for starch by adding iodine. It will obviously turn blue-black color, that means the food contains starch. Now, in another test tube, we're going to take the same sample, that is starch solution, and we're going to add equal amounts of saliva. I already told you that the saliva contains the enzyme salivary amylase. So your amylase, what it does is, it digests starch, converting into glucose. So now this sample, we are going to test for glucose and starch. It will give a negative test for starch. By this, what it means is, the starch has been digested by amylase. But this particular sample will test positive for glucose, which means the starch has been digested into glucose. So this proves that amylase digests carbohydrates, which is starch, converts into glucose. Next experiment is to determine the action of enzyme Pepsin and hydrochloric acid. Pepsin, you all know, are enzymes which acts on proteins, breaking it down into peptides. So what happens is, we're going to take 4 test tubes, label them as A, B, C and D. Then the given food sample, we're going to boil it. Let's say we're going to take albumin of egg. We're going to boil it and then filter it. When we filter it, we'll obtain a cloudy filtrate. This cloudy filtrate we're going to add in the test tubes A, B, C and D. In test tube A, we're going to add hydrochloric acid only. In test tube B, we're going to add pepsin only. In test tube C, we're going to add pepsin and hydrochloric acid. In test tube D, we are going to add boiled pepsin and hydrochloric acid. Now, out of these 4 test tubes, only in test tube C, the cloudy white appearance disappears. That means the protein is being digested by pepsin, but not in the other 3 test tubes, that is A, B and D. Now you might be asking, in B we have added pepsin, so why no digestion? Now remember, in stomach I already told you that the food mixes with the gastric juice. Gastric juice contains hydrochloric acid. This hydrochloric acid converts inactive pepsin into active pepsin. Only your active pepsin is capable of digesting proteins. So this is the explanation for cloudy white appearance not disappearing in test tube B. Again, you might argue why not in B, but in D what happens is we are using boiled pepsin, and pepsins are nothing but enzymes which get destroyed upon heating. So since we are using boiled pepsin, that means this pepsin has been destroyed. So this experiment proves the effect of hydrochloric acid and pepsin. Hydrochloric acid, what it does is it activates the pepsin. Pepsin in turn digests proteins. So this is a simple experiment to determine the action of pepsin and hydrochloric acid. Similarly, another such experiment is to determine the effect of acidity and alkalinity on food. Basically, the moment your food reaches the stomach, it becomes acidic because your gastric juice contains hydrochloric acid. Now, too much of acidity is also not nice. So, this acidity is reduced by bile and pancreatic juice in the stomach. That means the food now again becomes more alkaline. So, in this particular experiment, again we are going to say take the same 4 test tubes, but we are going to add another test tube and label it as E. Again we are going to take the same sample, that is boiled albumin filtered, which is nothing but cloudy white appearance. Again the same thing. In test tube A we are going to add hydrochloric acid, test tube B pepsin, test tube C pepsin and hydrochloric acid, test tube D boiled pepsin and hydrochloric acid, and in test tube E we are going to add sodium bicarbonate. Sodium bicarbonate is an base, that means it makes the food sample more alkaline. Now what happens is only in C the cloudy white appearance disappears, that means the pepsin gets digested, but in test tube E the protein is not digested because alkaline does not convert pepsin into active form. But alkalinity or basicity why is it necessary is, again in the small intestine what happens is fats is converted into emulsified fats, which is acted upon by lipase forming fatty acid and glycerol. So basically fats on itself cannot be digested by the enzyme lipase. It has to get converted into emulsified fats, and emulsified fats conversion is only possible when the food becomes alkaline, and this alkaline occurs in small intestine. So this is the end of the experiments.